because it can deaminate lysine and produce alpha keto carboxylic acid, which is responsible for the red color of the slant. Their negative is purple slant. So we have three possible LIA reaction, which is purple over purple. Let's start with purple over purple. Their lysine deaminase is negative. Their lysine decarboxylase is positive. Their slant is purple and their butt is purple. And now let's start with the purple over yellow, which their lysine deaminase is negative. Their lysine decarboxylase is negative also. Their slant is purple while their butt is yellow. Now let's go with the red over yellow. Their lysine deaminase is positive while their lysine decarboxylase is negative. Their slant is red and their butt is yellow. They only observed only among Proteus, Providentia, and Morganella. There is no such reaction as red over purple. This will explain that the enteric may either have lysine decarboxylase and produce purple over purple, or have lysine deaminase and produce red over yellow, or lack both enzyme and produce purple over yellow. However, the enteric organism will never have both lysine decarboxylase and lysine deaminase. Now, let's start with the control, the PY, and this. This indicates that the LIA can also detect H2S production. However, compared to the TSI, LIA is less sensitive for H2S production. Now, let's start with the review of the Mahon. Use of lysine iron agar. If detection of lysine deamination and lysine decarboxylation, which diff H2S production but results a variable reasons. Composition are the glucose lysines, which the slant where the amination happens aerobic. The pH indicator is the brom crystal purple. The butt where the carboxylation happens anaerobic. The H2S indicator is the ferric ammonium citrate, which is produced of alkaline product or cadaverine. Now the key over key, which is alkaline over alkaline, I guess. Yeah. Okay, let's start with our own. Purple over purple, which the LDC is positive. Example are these are the Klebsiella initial, which their initial is yellow, BEC of glucose form, which turns to purple, which BEC of cadaverine LDC positive. In the deaminase is positive, which an example of it is Proteus, which DIM positive to glucose positive remains yellow, which BEC it's not LDC, deaminate, which red slant. That's for red and yellow actually, RA. Sorry about that. In the deaminase. Now let's start with K over K, which is P over P with H2S production, which their CDC positive and H25. It's kind of hard actually to understand. So let's move on. So we have ornithine decarboxylation, which their medium is MIO, which motility, idle, and ornithine. They are semi-solid medium. There are glucose or ornithine, brom crystal purple. There are deep butt tubes. Their positive is purple butt, which is potricine, slight yellow tinge, and their negative is bright yellow butt with purple rim. Their motile is hazy medium, and their non-motile is clear medium growth in stub line. It can detect three properties of the enteric. It is the motility since it is semi-solid. It is the idle if you add Kovacs reagent after incubation, and the ornithine decarboxylation. With a wire needle, get the inoculum and stab butt once all the way down to the bottom. Then incubate. During the incubation, glucose is utilized producing acid. The MIO will turn into yellow. If positive, then the purple butt, potricine. Those which have the ornithine carboxylase will act on ornithine and produce putrescine, which is an alkaline product, reverting the bath into a purple color. There is a slight yellow tinge because of the acid from glucose. And their negative is bright yellow bath with a thin, narrow purple rim at the top of the medium. They're motile if the medium is hazy, and non-motile, growth confined only in the line of stub and on the top of the medium while the surrounding is clear. Then let's talk about the carboxylase, enzyme that removes the carboxylase group from amino acid, omnithin, lysine, and arginine. Lysine decarboxylase are the cadaverine production. Ornithine decarboxylase are the putrescine. The arginine dehydrolase are the citrulline, turns to ornithine, then turns to putrescine. Their medium is decarboxylase medium with brompencil purple, then glucose and amino acid medium overlaid with mineral oil. The result is purple, useful to differentiate Klebsiella from Enterobacter, the decarboxylase. Now let's go with the last actually, the malonate utilization. It detects whether the enteric can utilize malonate as the only source of carbon for metabolism. Medium, malonate broth. Their medium is malonate broth, which their bromthiamol blue as pH indicator, just like SCA. Get one loophole of this organism and suspend it on malonate broth, then incubate. Their positive is light blue to deep Prussian blue, due to formation of sodium hydroxide, which is the product responsible for the blue color. The enteric can utilize malonate as the only source of carbon for metabolism. Their negative is retain the green color. In summary, let's not discuss about this. Yes. Now, let's discuss about the opportunistic coliforms enteric pathogens. So, we have genus Escherichia is that under the genus Escherichia, we have Escherichia coli, which is the predominant facultatively anaerobic species in the large intestines of man. Index of fecal contamination in water bacteriology. Their AMB is greenish metallic sheen. Their in the MAC naman, there are flat, dry pink colonies. In the blood agar, some are beta hemolytic. Mostly strains are motile. There are lactose fermenters aerogenic. Their TSI is equal, don't know. Their IMBIC is equals to positive, negative, negative. Escherichia coli is the most common cause of UTI, non-nosocomial infection, and gram-negative bacteremia. E. coli on eosin methylene blue EMB agar, which they're blue, which they have background. Same, we don't need to, because this is only a voice. So colon Mancokia agar lactose fermentase versus non-lactose agar. We don't know about this. Okay, let's discuss this. There are two groups of pathogenic Escherichia coli. We have extraintestinal pathogens, which there is oropathogenic E. coli or OPEC, which are the major cause of E. coli associated community-acquired UTI. Causes cystitis and acute pyelonephritis. Their virulence factor is hemolysins, pili, cytotoxin necrosing factor, which an example of it is the E. coli 06K2H1. Their meningitis/sepsis. Then there's meningitis/sepsis. They are associated E. coli or MENEC causes neonatal meningitis that results in high morbidity and mortality. Their virulence factor is K1 antigen or 80%. Their E. coli is 07K1. Then we have the diarrheagic or interstitial pathogens, which there are 5 strains which can cause serious gastrointestinal tract infection. These are the enterotoxigenic E. coli or ETEC, Then we have enteroinvasive E. coli or IEC. We have the enterohemorrhagic E. coli or EHEC. We have the enteropathogenic E. coli or EPEC. And we have the enteroaggregative E. coli or AIEC, which transmitted by contaminated food and water. So, let's discuss here. In the ETEC naman, or the enterotoxigenetic E. coli, their path of mechanism is enterotoxin, LT, and or /ST. Their primary site is the small intestine, which their epidemiology is that they are travelers in childhood diarrhea. Their fever is absent actually, while their stool is copious watery. Their blood is absent, and the blood is absent, and the pus is absent. In the enteroinvasive E. coli naman, their pathological mechanism is that invasion of enterocytes. Their primary sites are the large intestine, and their epidemiology naman is common dysentery-like syndrome. In the fever, it is common, while in the stool, they are scanty, purulent, bloody. Their blood is common actually, and their pus is prominent. The EHEC, VTEC, the enterohemorrhagic E. coli or the VTEC or the STEC, their pathomechanism is virotoxin or Shiga-like toxin. Their primary sites in this is in the large intestine, which their epidemiology is that hemorrhagic colitis or hemolytic uremic syndrome or HAS. In the fever, they are absent, and in the stool, they are copious bloody. In the blood naman, they are prominent, and in the pus, they are absent. Then we have EPEC, or the enteropathogenic E. coli. Their pathway mechanism is that attachment and effacement of enterocytes, which their primary sites is small intestine. Their epidemiology naman is infantile and childhood diarrhea. Their fever naman is common, while their stool is copious watery. Their blood is absent, or their pus is minimal. Then let's go with the EHEC, which is enteroaggregative E. coli, which their path mechanism naman is adherence to enterocytes or AGGR. Their primary sites are unknown, which their epidemiology naman is that diarrhea in children like HIV infected and AIDS patients. That's their epidemiology. Their fever is occasional, their stool is watery, while their blood is absent and pus is absent. Then let's go with the laboratory DX, the ETEC or the enterotoxigenetic E. coli. It is a childhood and traveler's diarrhea. They are mild watery diarrhea with abdominal cramps and nausea and headache. Their LT and or ST enterotoxin, the LT enterotoxin, which is equals to heat label at 65 degrees Celsius for 30 minutes, they are antigenic. Their rabbit island loop test is positive to dilation of loop. In the Chinese hamster ovary cells assay, there is cell death. In reverse passive latex agglutination test, the DNA hybridization, that's the description of LT enterotoxin. Then we have the ST enterotoxin, which they are heat stable even at 100 degrees. Poorly antigenic. They are infant mouse assay and DNA hybridization. The enteroinvasive E. coli naman, it causes bloody diarrhea as a result of direct invasion of the enterocytes in the colon. An example of it is E. coli 0112, which serenity test is keratoconjunctivitis in 24 hours. Then we have EHEC or the enterohemorrhagic E. coli, which is they are virotoxin or cytotoxin with damages vascular endothelial cell causing hemorrhagic colitis. It can also lead to hemolytic uremic syndrome or HAS,